So what is the rationale for molecular targeted therapies in head and neck cancer? Historically, we've treated cancer with surgery, chemotherapy, and radiation, all of which are relatively nonspecific targeting therapies, if you will. And it's only by elucidating the targets in the malignancies that we can begin to identify molecules, proteins, transcription factors, etc., that could potentially be exploited for therapeutic purposes. And in general, if we take our tumors and we look at what is expressed, what is there in the tumors, and if the target is there, and in fact if it's there at increased levels in the tumor compared to normal tissue, that's a pretty good indirect indication that something could serve as a target. If we are able in cohorts of our patients through potentially immunohistochemical analyses, look at expression of the molecule and see whether or not it correlates with outcome, and Dr. Wolf gave us many examples of that this morning, and if indeed the factor correlates with survival, specifically decreased survival, that's another pretty good indicator of biologic potency. And perhaps the proof of principle is that when we go back from our patients to our models in the laboratory, cell lines, animal models, human tumor explants, and if we target that molecule with various strategies and we can essentially decrease the growth, increase the apoptosis, or eradicate the tumor in our models, that's also a pretty good clue that this might help our patients. And we did all of this with the EGF receptor. That's the good news. The bad news I'll get to very quickly. So what is the epidermal growth factor receptor? It's a very common receptor, type 1 tyrosine kinase receptor. It's overexpressed in many malignancies, including squamous cell carcinoma of the head and neck. It's stimulated by growth factors. The tumors essentially produce their own gasoline to drive the growth of the tumor in the form of autocrine ligands like transforming growth factor alpha and amphoregulin. And as I said, it's upregulated in cancers. But there's altered forms, and I'll tell you a little bit of a story today about how these altered forms, specifically the EGFR variant 3, that lacks a ligand-binding domain, may explain some of the disappointing results that we've seen in clinical studies. So there's several strategies available to target the EGF receptor. We're familiar with many of these. By far, the antibodies and the tyrosine kinase inhibitors are the most developed, but there's other ways to block the EGF receptor, and I'll tell you a little bit of a tale of how selecting a strategy that potentially has a different mechanism of action can really help elucidate the biology not only of this specific growth factor receptor, but our patients and their tumors, and how we can potentially optimize treatment responses using the same target. So as Dr. Wolf alluded to this morning, this paper really changed our lives. Based on this phase 3 trial that was supported by the Radiation Therapy Oncology Group, Cetuximab in combination with radiation was shown to significantly improve outcome. Barbara Burtness had led the trial in the ECOG that showed the addition of Cetuximab to chemotherapy potentially enhanced survival, but it's possible that that study was underpowered and didn't have the same impact in terms of changing the standard of care. A month after this paper was published, the FDA approved Cetuximab for the treatment of head and neck cancer, the first new drug for this cancer in the United States in 45 years, and I would say it really has completely changed our understanding of biology. It's frankly also, in the academic setting, changed the population of patients that we see, and I think introduced new challenges in understanding the biology of head and neck cancer and how we can treat this best. So to date, the Cetuximab is standard, and it's a standard approach in combination with radiation. There's emerging data in combination with chemoradiation, but I think that the fundamental issue with Cetuximab today is, number one, we don't really understand prospectively how to identify those patients who are more likely to respond to Cetuximab, and what's probably clear from all of the studies is that expression of EGF receptor, expression levels of EGF receptor in the primary tumor have really nothing independently to do with response to Cetuximab. The other thing that I think is still unclear, probably based on the paucity and the quality of the specimens from these phase 3 trials, is we still don't really know what Cetuximab does in human head and neck cancer. There are very few studies that have paired biopsy specimens, tumor specimens from patients before and after treatment, thus making it murky, if you will, how to determine what this drug is doing in the human head and neck cancer patient. EGFR tyrosine kinase inhibitors, gefitinib or erlotinib, have been heavily investigated in head and neck cancer. We don't have positive phase 3 data. We don't yet know how to use these drugs, but they're heavily investigated in clinical trials in this country and elsewhere. But the truth is that clinical responses to EGFR-targeted agents as monotherapy in most trials have been reported to be around 10%, which is unexpected given the 3 criteria that I started my talk with, all of which were met with EGF receptor, which led most of us to expect that the therapies would be far more effective. So what we'd like to really decide today and explore in all of our laboratories is how do we identify those patients more likely to respond to these drugs than other drugs, and in addition, how do we really rationally develop combinatorial molecular targeting strategies for individual patients that will improve their outcome. So I'm going to tell you a little bit of a story at the University of Pittsburgh in an oral cancer center study that was led by Gene Myers and Teresa Whiteside, where we took a different strategy to block the EGF receptor. And we took an antisense strategy. This was a long time ago. It was before there was siRNA. Probably today we would have done siRNA. But indeed, when we looked in our preclinical models and we compared the precursors to the antibodies, the precursors to the kinase inhibitors, and an antisense approach, which was designed to downregulate RNA and protein expression of the EGF receptor, we found that that was far more effective in our preclinical models. And so we designed a phase 1 trial. Phase 1 trials, as you know, are really for the purpose of detecting toxicity, but we were hopeful that with an intratumoral approach and the opportunity to obtain and interrogate specimens continuously on this study, that we would really be able to elucidate biology. We collaborated with the National Gene Vector Lab, which was then at the City of Hope, to manufacture the DNA. And then Stephen Lai, when he joined the faculty at the University of Pittsburgh, collaborated with me in administering the treatments, obtaining the biopsies, and he's the lead author on the paper that will hopefully appear in a journal near you someday. The biologic endpoints were EGFR expression levels and STAT signaling, and we used imaging to be able to assess response. And I'll show you some selected examples that I think will illuminate where we may begin to think about going with biologic agents and molecular targeted therapy as opposed to standard cytotoxic agents. So 17 subjects were treated. We treated once a week for 4 weeks. We went up to a very high dose of DNA. We used DNA without a virus, without a lipid carrier, otherwise known as naked DNA. And much to our surprise, 5 of these subjects experienced a response. And this is unexpected because the New England Journal reviewed all phase 1 trials in cancer about 4 or 5 years ago. The expected response rates in phase 1 trials for gene therapy was less than 10%. And as you can see, our response rates approach 30%. 2 patients' tumors disappeared completely. 1 patient recurred about 3 years later. Another patient died of myocardial infarction a year after the tumor resolved. And 3 patients' tumors decreased by more than 50% in maximum diameter. When we biopsy the tumor 6 weeks later, we could detect the DNA, the therapeutic DNA, in all of the tumors. And what we found was the amount of the gene didn't matter. What was most important for an intratumoral delivery method was the size of the tumor, and that's something that potentially would be expected. And in contrast to the earlier studies, and indeed the current studies today, as I just alluded to, using cetuximab and the tyrosine kinase inhibitors, is we did find that patients with higher baseline expression of EGF receptor were more likely to respond to the EGFR antisense gene therapy, and patients that had lower levels of the transcription factor STAT3, which in part is downstream of the EGF receptor, were more likely to respond. The other thing that was really curious and in fact gratifying was in virtually all of the patients where we had paired specimens for analysis, the EGFR antisense therapy did decrease the amount of EGFR expression in the tumor, which is what we would hypothesize. Those patients whose tumors shrunk had a greater decrease than patients whose tumors remained the same size or whose tumors progressed during therapy. And we learned something. We learned that there's patients like this individual here. You can see they have a large metastatic lymph node adjacent to their prior tracheal stoma. It's very FDG avid. This is a PET CT and an independent CT pre-treatment. This is post-treatment. This is 6 weeks later, 4 injections of the gene. The size is exactly the same. So technically, this is not a response. But the SUV, the standard uptake value of the FDG glucose, is dramatically downregulated. This patient did not want any further therapy. He went home. He lived 9 months, which is unexpected in this patient that's heavily pretreated with recurrent metastatic disease. And so it's possible that with these newer agents that aren't necessarily cytotoxic, that surrogate markers of activity, PET markers of metabolism, of apoptosis, of hypoxia, of proliferation, may be more useful in evaluating response to therapy than standard anatomic imaging. And by immunohistochemistry, this is what I alluded to, what we found is that pre-treatment, EGF receptor is the brown staining, post-treatment, EGF receptor was downregulated. It sounds so trivial, but it had never really consistently been reported using an EGFR targeting agents in cancer patients that the agent had really blocked something biologically inherent to the activity of the receptor, in this case, protein expression. And this is just the cumulative results showing that higher baseline EGF receptor predicted response, and we also found that lower STAT3. Now, the real fundamental question I think facing us today, since this is really our only approved molecular targeting agent, is why aren't all of our patients responding to it? Well, I think we don't really understand the dependence on EGF receptor, which is also an oncogene. So oncogene addiction is a common biologic term. Very few of us really understand what it means, but in essence, it means that it's necessary and sufficient, that the EGF receptor is present in the tumor, and that's really driving the growth and resistance to therapy programs in that tumor. It's quite possible, as Dr. Wolf alluded to earlier this morning, that EGF receptor is just one of the many targets that are important for a patient's tumor, and trying to sort out in each individual patient the relative dependence on EGFR versus other targets, I think, is important. And what we're trying to do today in our laboratories, and what I'd like to spend the last few minutes on, is really understand the mechanisms of resistance to EGF receptor targeting, mechanisms of escape from EGFR blockade. And our lead theory today is that it's not just the EGF receptor. I mean, we get grants and published papers being very myopic and focusing on a single pathway, but in reality, there's lots going on in our cancer cells. This is from a review article that we published with Aline Forestier's leadership in the Journal of Clinical Oncology a couple of years ago when the issue was devoted to head and neck cancer. And as you can see, there's so many pathways going on. There's many opportunities for crosstalk at the receptor level, in the cytoplasm, in the nucleus. And if we just block one pathway, particularly up here or up here, there's so many other circuitous routes to go downstream and to drive tumor growth pathways that it really behooves us to try to elucidate the specific program and eventually get to personalized medicine. So one of the stories that we've developed is STAT3 as a therapeutic target. Now, this is a story that I'm going to tell you that's been very challenging because unlike the EGF receptor, where there's many now large pharmaceutical companies that have made agents that selectively block the EGF receptor that we can use, to date, there's no selective STAT3 inhibitor that's been available or tested in any human being. So we made our own. So this is a story about how we made our own. So STAT3 has been associated with transformation of epithelial cells, of fibroblasts. Constitutive activation of STAT3 tyrosine phosphorylation is detected in head and neck cancers and many other malignancies. And most importantly, I'll skip through to the end, is we found that in our cell lines that EGFR targeting resistance was accompanied by increased activation of STAT3. So we thought, maybe if we block STAT3 in conjunction with upstream inhibition of the receptor, we can essentially overcome that resistance. And other groups from around the world, particularly Bernie Weinstein's group at Columbia, has reported that phosphotyrosine STAT3 levels in the primary tumor are independently correlated with decreased survival in head and neck cancer. So, what have people done today to block STAT3? There's a whole series of small molecules, many of which are natural products that have anti-STAT3 activity. These also tend to be NF-kappa B inhibitors. They're not particularly selective, but they do have some activity against STAT3, and they are in clinical investigation, and we're working collaboratively with many groups at Emory University, at MD Anderson, and others to try to develop these for further clinical application. What we know in head and neck cancer is that to get to STAT3, you have three possible options. There's the EGF receptor, which we've already talked about. There's Sarc family kinases, and work by Silvio Gutkin and others have shown that IL-6 GP130 is another way to get to STAT3. So if you're going to block upstream receptor and non-receptor kinases, you got to have a cocktail. It's got to be all three. So we used a transcription factor decoy approach, which essentially is a double-stranded piece of DNA. It's to trick the cell that STAT3 is signaling. The decoy, if you will, binds to the STAT3 response elements and the promoters of genes and essentially stops STAT3-mediated gene expression. And because there was no STAT3 inhibitor and we're not a drug company, we applied to a wonderful program through the NCI called the RAID program, or Rapid Access to Industrial Development. And what they did is they provided the support to manufacture the clinical grade DNA, which is now at the University of Pittsburgh. We're about to open the study. And they also paid for and oversaw all of the preclinical toxicology experiments that were required by the FDA to get our IND. This is also something that was really beyond our expertise as an academic laboratory. And so this is what our decoy does in preclinical models. When you treat head and neck tumors with this transcription factor decoy, you can decrease the tumor volume. It's important that in all of these experiments, we've used a mutant control decoy that's also a double-stranded piece of DNA, differs by one base pair, doesn't bind STAT3, and it virtually has no effect on tumor growth. And what's extraordinarily gratifying is when other people take your compound and they not only reproduce what you've shown, but they go much farther. And so this is work by John DiGiovanni's group at MD Anderson using his mouse models of squamous cell carcinogenesis. And what John and his group reported several years ago is that using our same STAT3 decoy, when they injected the decoy into the lesions in the mice, they could essentially eradicate the squamous cell carcinomas compared to the mutant control. Our decoy has now also been reported in studies of breast cancer, lung cancer, and now glioma. So I think the beauty of head and neck cancer and the opportunity that we have as head and neck cancer investigators is to use head and neck cancer as a model, because our tumors and our patients are relatively accessible for biopsy and interrogation, and only the head and neck surgeon can do this. So even if we don't run our own laboratories, let's all be professional collaborators, and let's work with the team that Greg mentioned, because we're essential. And then if we form these principles in head and neck cancer, the far-ranging implications of our findings go beyond squamous cell carcinoma of the head and neck to other epithelial malignancies. So this is design of our Decoy Trial, which just opened in Pittsburgh, and it will be led by Dr. Steve Kim, who you heard from Steve Lai earlier, is also working in our group in Pittsburgh. What we're going to take is patients who are having surgery for their head and neck cancer. When they're asleep for their tumor resection, we will take a biopsy, and then we will inject the decoy into the tumor. They'll have their operation, and then the specimen will come out. So the end point of the trial is biology. So this is a phase 0 trial, a concept introduced by the FDA in 2006 to try to really look at pharmacodynamic endpoints of agents that may or may not be likely to be developed clinically. We have some reservations about intratumoral administration. We think it's unlikely to go beyond the early clinical trial setting. So what our hope is that if we can demonstrate that the STAT3 decoy blocks STAT3 target genes, then in collaboration with chemists at Carnegie Mellon University, we've made some modifications to the decoy that seem to prolong its half-life in serum and in stability, and hopefully this can be administered systemically in a subsequent study. But it's not so simple. I wish it was just EGFR and STAT3, we'd be done. But it's not. There's other growth factors, tyrosine kinase receptors, non-receptors that are involved. And so in the Sarc-centric view of the world, you can see that not only do receptor tyrosine kinase, but G protein couple receptors and other components of the extracellular compartment signal through Sarc family kinases to mediate growth. And these are two inhibitors that are either FDA approved or in clinical development and are being actively studied in cancer patients, including head and neck cancer, trials ongoing at Memorial and at MD Anderson, and I'll tell you a few of the studies that we're doing at the University of Pittsburgh and through the SPORE mechanism with several other institutions. So in contrast to the EGFR antisense or the STAT3 decoy, where we made it ourselves, which is sort of nutty for a surgeon to do that, at least in this particular case, we could take advantage of safe agents that have been developed by pharmaceutical companies. And so what we do, and I think this is important as a head and neck cancer surgeon scientist, is we can't spend all of our time in the laboratory interrogating preclinical models because then it loses relevance for our patients. So what we'd like to do is develop models where we go back and forth very rapidly, so translational science going both ways. So once we had all of this preclinical data that SARC mattered, we went back to our patients. In collaboration with Gordon Mills at MD Anderson, we did reverse phase tissue microarray. This was led by Anne-Marie Egloff in our group, and we looked at expression levels of CSARC and expression levels of EGFR in the head and neck tumors and the adjacent normal mucosa, which we know is at risk for subsequent tumor formation. And in both of these tissues, we found that CSARC and EGFR were highly correlated with each other, which led us to believe that there might be some benefit to think about co-targeting strategies. We then used dasatinib, which is the FDA-approved agent. It's approved for leukemia and lymphoma, and we treated head and neck bearing xenografts with dasatinib and found a dose-dependent inhibition of tumor growth using doses that were comparable to what we're giving to people. So this seemed like a feasible strategy to use. I just want to emphasize it's not just one drug. We also looked at the AstraZeneca inhibitor, AZD05130, as well as gefitinib, and this is work done by Sung-Ho Choi from Korea, who's in this audience, who did a postdoc in our laboratory. And indeed, combination of these two EGFR and SARC inhibitors additively or synergistically inhibited growth as well as invasion in our preclinical models. So this is a study. So you're going to now begin to see a theme where I think that we can really, as surgeons, contribute to research and study every patient. So here's a study that we're going to do with the head and neck spores and lung spores. This is supported by the NCI as well as both pharmaceutical companies that make the agents, which is a bit of a political challenge, but was very feasible and doable. So the study will go on at Vanderbilt, at Pittsburgh, at Emory, at Anderson, and at the University of Colorado. We're going to take patients who have head, neck, and lung cancer. So another message is that there's so much similar biology in squamous cell carcinoma of the head and neck and squamous cell carcinoma of the lung. So let's partner with our air digestive tract investigators and try to do studies that are feasible together, because lung cancer is a much bigger disease. There's more opportunities for funding, and I think we can work together and not against each other. So we'll take patients who are having surgery for head, neck, or lung cancer. We'll randomize them to receive an EGFR inhibitor or erlotinib, dasatinib, combination. We'll also have a small placebo group. We'll give them this treatment while they're waiting for their surgery a couple of weeks. Then we'll get an imaging study, and there's a recent report from a group in France to show that 2 to 3 weeks of erlotinib resulted in 30% of the patients having a measurable decrease in tumor volume. So we're hopeful that we'll see some surrogate clinical activity using the anatomic imaging study, and then we'll harvest the specimen. We'll look for the primary endpoints as well as proliferation apoptosis. But what we'd really like to do is build on what Dr. Wolf suggested, is do comprehensive genomic and proteomic analysis of the specimens when appropriate, pre and post-treatment, to ultimately try to identify how do we use these agents in a therapeutic setting. Because even though all of the tumors are squamous cell carcinoma under the microscope, we know that they're not all driven by EGFR and or SARC pathways. Very quickly, I'll tell you about mutations in EGF receptor, which I actually don't think exist in head and neck cancer to date. Many have sequenced the tyrosine kinase domain. This has been reported in selected examples of non-small cell lung cancer. And I think with the exception of 5 male Korean smokers, there's no head and neck cancer patients that have TK mutations. So I think it's not going to be a primary modality of EGF receptor alteration. But we reported a couple of years ago that this variant 3 is detected in 40% of head and neck cancers. We looked at another cohort from Vanderbilt and found indeed it was about 38% of the 88 tumors that were sent to us by Christine Chung and Del Yarborough. And this has also been validated in the University of Chicago cohort, who did the same kinds of studies. So this is a truncated version of the wild-type receptor. And the really important story is that antibodies like cetuximab don't bind very well to receptors that don't have an external ligand binding domain. So we think this represents a really plausible mechanism of resistance to cetuximab because the cells that express this altered receptor are not growth inhibited. And we have extensive preclinical data showing that, and Shinsuke Suzuki from my lab will present that data later in this conference. But very quickly, if we look for L8A4, which is the EGFR V3 specific antibody, we can detect it. The bottom line is it's always there with wild type receptor. There's no such thing as a V3 pure head and neck cancer, so there's V3 and wild type together. The mechanism of V3 to alteration remains elusive, and we're trying to distinguish DNA alterations, that is microamplifications and microdeletions, from splice variant or RNA editing. But that's tricky to do, but hopefully we'll have an answer in about a year for you. But the important thing is that when you put this altered receptor into head and neck cancer cells, the tumors grow dramatically and compared to wild type receptor. So here's just some mice. Here's the V3 expressing tumor on one flank, the wild type receptor on the other. And in all of the cases, we saw dramatic growth enhancement with V3. And the growth enhancement was resistant to cetuximab. We see increased invasion. Increased invasion is also resistant to cetuximab. And so we think that this is very plausibly a mechanism of resistance in head and neck cancer patients. How do you block EGFR V3? Well, there's a toxin developed by the NCI RAID program in collaboration with investigators at Duke that are now testing this in glioblastoma. They're very interested in working with us. In fact, I just got an email from Daryl Bigner, who's the PI of the brain spore at Duke. They'd like to do this in head and neck cancer, so those of you who are interested and you'd like to collaborate on a phase 1 setting where it's intratumoral administration of the toxin, just let me know or contact Daryl independently. It should be fairly trivial to cross-reference their IND. There's an antibody that has special affinity for the V3 because there's a novel glycine residue at that truncation that they created an antibody against. This was made by Ludwig. Ludwig has just licensed this or in the process of licensing this to Big Pharma, and a formal phase 1 trial is planned. I'll show you some results in head and neck cancer patients from a pilot study. And for reasons that none of us really understand, these irreversible inhibitors, HKI-272, Boehringer Ingelheim also has a compound. They irreversibly inhibit EGF receptor and HER2, but they also appear to have selective activity against V3, and the group at the University of Chicago is actively investigating these agents in head and neck cancer patients. So this is some data from our group using the MR11 toxin. It does inhibit the growth of cells engineered to express EGFR V3. And this is some work shared with me by Terry Johns at Ludwig from their PNAS paper about a year ago. You can see that this is a head and neck cancer patient, and the antibody, which has been labeled with indium, is actively taken up into the head and neck tumor. So there could be some promise with this agent as well. So in summary, I'd like to this is just a bit of a story. There's many other stories under active investigation, but if we really interrogate our patient's tumor, either in our own laboratories or in partnership with other laboratories, we really will begin to understand what's different about our patient's tumors compared to normal tissue and potentially other cancers. There's several targeted therapies that are in early clinical development in head and neck cancer. I talked today about SRC, STAT3, and V3. Also, c-MET targeting is becoming increasingly plausible because c-MET appears to be a mechanism of resistance to EGFR TKI in lung cancer, and work by Paul Harari's lab has shown that this is also a mechanism of resistance to cetuximab in head and neck preclinical models. mTOR targeting is under active investigation by many groups, particularly Cherry Ann Nathan's group, and IGF-1 receptor targeting using antibodies or kinase inhibitors is also under investigation primarily by the group at MD Anderson. But I think our ultimate goal is how do we profile our patients' tumors and then really rationally select these agents that are increasingly being developed and will be on our pharmaceutical shelf to treat individual patients. So I was very fortunate this year with the support of Ki Hong to get some resources from the American Cancer Society. And as an American Cancer Society clinical research professor, what we were trying to do is enable every head and neck cancer doctor, every head and neck surgeon to study every head and neck cancer patient. So what we're doing in Pittsburgh is setting up an engine so the patients who are having surgery for their head and neck cancer will be randomized to receive a candidate molecular targeting agent that's been proven safe in phase 1 trials. We'll also have small groups receiving placebos because all these analytic platforms are only as good as your controls. And then we will image the patients very similar to the SPORE study I described to you earlier to have a surrogate clinical endpoint. And then at the time of surgery, we'll harvest the specimen and partner with all of our collaborators to truly understand the pharmacodynamic properties of the agents in our cancer patients so that we ultimately can design more rational care for our cancer patients. And so I'd like to conclude by thanking everybody here, past and former and current trainees, the head and neck program, and in my laboratory, as well as all the collaborators elsewhere. Thank you very much.